Okay, so this then is lecture 8. And in the last class, we introduced amino acids. And of course, they're named for the fact that they have this amino group here. And then they have an acidic group, which in this case is deprotonated from a carboxylic acid. At physiological pH, it will deprotonate to a carboxyl group, and so this carries a positive and a negative charge. Depending on what the R group is, that determines which of the 20 amino acids we're talking about. And if that R group is anything other than a hydrogen, which would represent the amino acid glycine, if it's anything other than a hydrogen, then there's going to be stereochemistry around that alpha carbon. And this means there are two possible configurations. And if we think back to chemistry, we would like to, anytime we see a stereocenter, we like to assign it as R or S based on this whole set of rules of prioritization about the carbon. However, when we're looking at amino acids, we don't do that. And so I'm just going to reiterate this point because it's actually quite important. We utilize what's called an absolute configuration. And what I did was I made a little slide to show you what that actually means. The top of this figure shows amino acids in the L and the D conformation. And below it, I can show you the molecule that we actually compare them to. So in absolute configuration, we're comparing our structure to that of a known structure. And in this case, we're looking at the L and D isomers of a sugar called glyceraldehyde. And these D and L isomers of glyceraldehyde are also, I should note, these are also the R and S forms if we're utilizing proper chemical terminology. What we can see is that this is the structure of glyceraldehyde. It's got a carbon, a central carbon, and then here there's a carbon attached to a hydroxyl group, so there's an alcohol. There's an aldehyde, so what this looks like is C double bond OH. I've just represented that as CHO. And then here we have OH. So this is the highest priority group, followed by this, followed by that, and of course hydrogen's in the back. So we can see that it's going to the left for lever rotatory or sinistra, depending on which we're using. All we do to use this as an absolute configuration is we swap out this group, the alcohol, for the R group. We swap out the carboxyl for the aldehyde, and we swap out the hydroxyl for the amino. And this can actually be done chemically through a small number of reactions, but this is why we use this configuration. And the reason we care about this configuration, remember, is that it doesn't matter what the R group is in this case. The R group can be anything apart from hydrogen, and if it's in the configuration to the top left, it's the L isomer. If it's in the top right, it's the D isomer. And we care so much about this, I'm sure you do, because ribosomes, which are the machine that makes proteins based on an mRNA message, the ribosomes for all life, all organisms, will only recognize the L isomer. Okay? There are examples of D-amino acids. They're commonly used by bacteria to make a protective semi-protein coat around themselves, but those assemblies using D-amino acids are not done by ribosomes because ribosomes won't recognize the D isomer. Yes?
Why don't the ribosomes recognize the D isomer?
It's actually not going to be the ribosomes. It's actually going to be the synthetase, which adds the amino acid onto the tRNA, but I'm just kind of ignoring that because we didn't go through the process of translation. Yeah, and so it's just structural limitations prevent the D isomers from being recognized. Because they're actually, if we imagine the R group is something other than just a little letter R, that's quite structurally very different. And we learned last class, change structure, change function. Okay, so hopefully that clears up the idea, this concept here. I can post this as an additional slide on the course space, or sorry, we're using Brightspace now. Okay, and if that isn't up by the end of the day, maybe someone can send me a reminder and I'll post this additional slide. We talked about the 20 amino acids, and we really, at this point, haven't gone into a lot of details. And that's kind of purposeful. We will cover that if you take 299 or Biochem 300A. We'll talk about the specifics of amino acids as they arise. And for example, we're going to look specifically at one today. All we did was kind of memorize them in groups. And we group these by nonpolar or hydrophobic residues. And we know that we're going to try, that the system will attempt to limit the interaction between a hydrophobic amino acid and an aqueous environment. And then we looked at the polar charged and uncharged residues, which interact favorably with the aqueous environment. And finally, we talked about the peptide bond. So the peptide bond, shown here, is the product of a condensation reaction. The way these are actually built is they're built in this direction. So we start at one end, and this will become what's known as the N-terminus for amino terminus. And then another amino acid is added to the C-terminal end. And then the next amino acid will come in at, oops, what have I done? The next amino acid will be added at the far right after that one that's an alanine, and so on until we produce a linear molecule. And the original molecule that's made by the ribosome will be linear unbranched. Modifications can occur to proteins after they're made, and we'll discuss those later in the course. So this peptide bond actually has a bit of double bond characteristic. So it doesn't rotate, it doesn't, and that means that that part that's sort of shaded in the purple, we can think of that as behaving as a planar entity. So it's like a plate or a sheet of something. And it won't rotate within itself, so it won't twist, but it can rotate at those two corners that are attached to, that's supposed to be an alpha, these two alpha carbons. Okay. So today I'm going to give you some more important exam information. You're probably getting tired of important exam information. We're going to talk about primary, secondary, and tertiary structure, and maybe go into a little bit of the section after that, which is called domains. Domains take quite a while to explain, so we may start them today, or we may, but we'll probably need to finish them on Tuesday's lecture. So here's the sort of important note about the exam. So on the syllabus, it says the exam will go to the end of 2A. I would like you to have as much time to study as possible. And I know next week on Thursday, a lot of people will be busy for Truth and Reconciliation Day, so that you kind of lose a day of studying there. So what I'm going to do is I'm going to actually cut it kind of in the middle of the protein section. And we're going to cut it right after this slide. RNA is a structural example. We should get that far today, but we may have to pick up a little bit on Tuesday, depending on how quickly we move through the material today. Okay? So I just want to make sure everybody knows. You can take a picture of this maybe so you're reminded. But what we have is it's going to cut off at this slide. And I've altered the syllabus, and I'll post the new copy of the syllabus today. Okay? And so what that means is domains, which we may or may not get to today, domains are actually going to be on the second midterm. So we're kind of not rushing our way through the material this year, and so I'm sort of half a lecture behind where I might usually be. Okay, is there any questions about that? Yes?
I just have a question about your office hours. I couldn't find the Zoom link.
There they are. Ta-da!
But where is the Zoom link? That's my question. Is I
It's just the Brightspace link. There, if you go on Brightspace, there's just a link. It says like Zoom link in office hours or something like that.
Like, is there a folder or something? I looked at it.
It's just in the normal, should just be in the normal file. Let's see if I can get online here. Two seconds here. Okay, so if you go into the site and you just click on content and you look under lecture materials, you'll see Zoom links here.
So that is the office hour one?
That's your link, yeah. That's the link for everything. Yeah, okay? Any other questions? Okay, great. All right, so I'll have extra office hours Wednesday at these times, 11:30 to 12:30 and 2:30 to 3:30. Okay, but I won't have the usual Monday hours because on Monday I have another exam and I don't want students kind of coming in and me accidentally answering exam questions for them because there is some overlap in the material. Okay. All right. So amino acids are put together and they form a chain, a linear chain called a polypeptide or a protein. We're using those terms interchangeably. We talked about that in the last class. There can be a number of interactions between the amino acids, and some of them are shown here. And in this example, we would call these intramolecular interactions because it's within a molecule. If it was interactions between two proteins, we'd call it intermolecular reactions. The first one here is showing a disulfide bond. If you have a cysteine, the amino acid cysteine looks like this. However, under oxidizing conditions, if there's another cysteine close by, they will form what's called a disulfide bond. And this only happens under oxidizing conditions. Okay? So we oxidize this to a disulfide bond. And that's a covalent bond, which is very strong, remember. And we don't expect this to easily break. Under reducing conditions, electrons will be donated to these, and instead of looking like this, we'll be back to having two sulfhydryl groups like this. So that's a disulfide bond, and that's the first example of an amino acid where we've discussed its function. So cysteines then can form intramolecular covalent bonds, but also intermolecular. If there's another cysteine residue nearby on an adjacent protein, so a second protein, those can form disulfide bonds. Oh, sorry, there's more on that slide. Here is an example of hydrogen bonding, okay, between an aspartate and a serine. Here we have ionic bonding, so the positive and a negative charge between an aspartate and a lysine here. So this is on the lysine and this is on the aspartate. And then at the bottom we have three hydrophobic residues, and we're seeing how they can actually have van der Waals interactions there. And so these are intramolecular interactions, and these will help to fold this thing into a three-dimensional entity. And remember that structure, the three-dimensional structure of that protein determines its function. Okay? We talk about protein architecture in a sort of hierarchical or in levels. The first is the primary structure. And this is the amino acid sequence, the linear order of amino acids. And what determines the linear order of the amino acids? What do you think? What's going to determine the order of those amino acids? The sequence of the RNA, which is related to the sequence of the DNA. So the sequence of a gene on the DNA determines the primary sequence. And the primary sequence we can call the primary structure, and that's just the end-to-end amino acids that are added into a chain that's linear. It's not branched at this point.
Sorry, can you repeat that?
It's linear. So
Sequencing thing.
the sequence of the DNA determines the order of amino acids. Yeah. So the sequence of a gene determines the order of the amino acids. Secondary structure are these small, regularly repeated structural elements. And we're going to go into quite a bit of detail about these in a minute. So I'm not going to talk about it much here. The tertiary structure, that's the fully folded protein if it's a single protein. So the way a single protein folds in space is called the tertiary structure. And quaternary structure occurs when there's two or more proteins. And that's the full complex of all of the proteins. It might be 2, it might be 3, it might be 80, whatever it is. That forms quaternary structure. And this is just a description, table 3.3 of your textbook talks about it a little bit. If we're visualizing this in space, to the top left, we can visualize the primary structure, which is just the linear chain of the polypeptides. The next slide, we have an example here of what's called an alpha helix, which is an example of secondary structure. That alpha helical component, and so we can see the linear structure within there. To make a secondary, we can see the secondary structure makes up a part of the tertiary structure. And the tertiary structure, this is a single polypeptide, a single protein. And that single protein here shown in blue is part of a larger complex. It looks like maybe there's 3 proteins overall in this, whatever it is. This is now our quaternary structure, which includes the other proteins as well. This slide then is representing the primary sequence. This is an example taken from the textbook. What we can see here are 2 separate polypeptides. And what we're looking at is the linear sequence of them. This is also showing disulfide bonds in here. So we can see that this cysteine and this cysteine can form a disulfide bond, which means that actually in physical space, they're very close to each other. Because remember, the distance of a covalent bond is about 1.5 angstroms or so. So those have to be very close to each other in the folded form. This is also showing some intramolecular disulfide bonds. Whether disulfide bonds are part of the primary, secondary, or tertiary structure, people can argue over that, and you're welcome to do so, but I'm not going to worry about it. As far as this textbook is concerned, they call disulfide bonds a part of the primary sequence, we can go with that, and I won't be concerned. I'm also not going to ask you about that. So there are times, I would say here, that the ones between the two peptides would actually be a part of the tertiary structure. You could argue for that, or the quaternary structure. But I don't want to get bogged down in those kinds of details, so we're going to just pretend it's not happening. Okay, secondary structure is weird. Remember back when we were talking about hydrogen bonding, and I showed a little picture. One of the things had a peptide bond. And if we go back and look at our peptide bond, what do we see about the peptide bond? Well, right here, actually let me erase this. Right here, it's got a great hydrogen bond acceptor, and here it's got a great hydrogen bond donor. We know that there can't be twisting of that peptide bond, so you can't get hydrogen bonding within a peptide bond. So that hydrogen can't pop out towards you and the oxygen pop out towards you and form a disulfide bond. Make sure you are totally clear on that. But if there's another peptide bond nearby, we can get hydrogen bonding between peptide bonds. And what was noted was that, in fact, what we start to observe is hydrogen bonding amongst the peptide bonds. And for some of these features, they're regularly repeated hydrogen bonds. And so I have a great example that will show you what I mean by that. And I'm going to pop 2 ahead to this. So this secondary structure is called an alpha helix. And if we look at the peptide bonds that form, it's between the oxygen, the carbonyl, oxygen here, and the residue 1, 2, 3, 4 away. Then the next one, 1, 2, 3, 4 away. Then this one will be 1, 2, 3, 4 away. And so we get a repeated pattern of hydrogen bonding. And what that results in is formation of this structure, and we call these secondary structures. Okay? So secondary structures are held together or formed or maintained by hydrogen bonding along the peptide backbone. The side chains can contribute to the stability or instability of these, but the actual secondary structure is held together by hydrogen bonding. Okay, I'm going to take a 2-second break here because I think it's 5:02. If anyone's going to head out, now is your chance. I think a few people might. Good luck. So we'll just take a 2-minute break here. Sorry?
Chances any paper towel up there?
Sorry. Yeah. Okay, so let's start up again. And again, if anyone's leaving for this, they can watch the recording later, so I'll post that this afternoon. So what we have here then is this structure called an alpha helix. And so it's a corkscrewing structure. But what's important, and it's maybe obvious from this slide or maybe not, but what we have is that the side chains of the amino acids are actually sticking out from this. So if we were to turn this, so you're looking at it down the axis, so it would look kind of like a circle, we would see that the side chains stick out. Okay, they project outwards. So they can interact with other residues. They can interact with each other. If you, say, had 2 of these right next to each other and they're really bulky amino acids, so branched ones, that might destabilize your alpha helix a little bit. Okay. Oh, but if you had a plus and a minus charge, that might stabilize it. So as I said, the R groups can affect the stability of the secondary structure, but the core component holding it together are these hydrogen bonds along the peptide backbone. So that's an alpha helix. A beta sheet works when you have, so an alpha helix, it happens within a discrete small section of the primary structure. For a beta sheet, we actually have to have two separate parts of the primary structure, and they come together as strands. And so a beta sheet has multiple beta strands that make it up. And they can be a huge number of strands or just a couple of strands. And the thing about the strands is that the peptide bonds, remember they're planar? Who remembers that? Everybody? Good work. So they're planar, and what happens with those peptide bonds is they're lying flat together in a plane, and they're hydrogen bonding across each other. And so you have, the plane is sort of shown here. Here's one planar part here. Sorry, it's hard to draw and see. And then here's the next one. And it actually ends up making kind of a crinkly sheet. It's not perfectly flat. And it's very hard to describe. So I have this video that I'm going to attempt to show you. And I'll post the video on Brightspace today as well. And this does a better job of showing us what these different secondary structural features look like.
This is a cartoon representation of the structure of human protein mRAS, a member of an important family of small GTPases. For reference, the PDB entry.
So in this, the beta sheets are shown in red, and the alpha helices are shown in the cyan blue color. Okay? So we can, and I'll talk a little bit about how these are depicted in our Jane Richardson slide. Okay.
For this structure is 1X, 1R. Now we will highlight a single beta strand as a cartoon ribbon with the backbone atoms as sticks and the side chain atoms as lines. Notice how the side chains stick out of the wide, flat sides of the ribbon, while the backbone amide hydrogen and carbonyl oxygen atoms stick out of the narrow edge. When we put this single strand in the context of a parallel beta sheet structure, we can highlight the hydrogen bonds stabilizing the sheet. These appear as dashed lines. In the parallel sheet topology, the hydrogen bonds may appear to zigzag a little because they are slightly out of plane. But two adjacent strands of this sheet are anti-parallel to each other, and when we examine their hydrogen bonding pattern, we may notice that the hydrogen bonds appear slightly more even and straight. In anti-parallel sheets, the hydrogen bonding pattern can be planar. Both parallel and anti-parallel sheets have the same number of hydrogen bonds per amino acid. This structure illustrates how a single beta sheet can have both parallel and anti-parallel portions. The strands of the sheet have an overall twist such that the entire sheet is shaped like a saddle, sitting between other alpha helical portions of the larger protein.
Okay, so that introduced another term there where they talked about parallel and anti-parallel beta sheets. So I'm going to draw us what we mean by that. So we know that proteins are linear. They have an N-terminus and a C-terminus, and that's how they're put together. So the way a beta sheet can work is that it can form this way, so that, and what we can do is we can draw the directionality by overlaying a big arrow here, and the arrow always points to the C-terminus. Wow, I'm having a really hard time drawing an arrow. Shocking. And then we can see, and then here this arrow points this way. So we can see that this is what we call anti-parallel, because they're pointing in the opposite directions, are really well-drawn arrows. But we could also have an example like this, where now our arrows are actually pointing in the same direction, and we call those parallel. Okay? And so they can form either parallel or anti-parallel sheets. They do look a little bit different, and that was alluded to in the video there, but we're not going to get super concerned about the differences. Keep in mind, those sheets are held together by hydrogen bonding with the adjacent peptide bonds. So secondary structure is kind of the second level, and then the final folded structure we call tertiary. And the tertiary structure will usually include elements of secondary structure, although there are proteins that lack secondary structures but still have tertiary structure. So this is the overall folded shape of a single polypeptide. And here's a couple of examples. If you look at the tobacco mosaic virus coat protein, it's mostly alpha helical. The central is showing this an immunoglobulin, so that's an antibody, part of an antibody. We can see that it's made up entirely of beta sheets. And then on the right, we have a kinase. These add a group called a phosphoryl group to substrates, and we can see that it's a pretty good mix of beta sheet and alpha helix. And so what we can see from that is the value of those diagrams that have the arrows and the alpha helices shown as they are. And this type of diagram was made by a structural biologist called Jane Richardson. We can call these Richardson diagrams, sometimes you'll see them referred to as ribbon or cartoons, but it's a really great way to represent then a protein. And actually, because this is what I'm going to do is something so exciting. Hopefully, wait for it. Wait for it. Okay. Okay, so what I'm going to do is I'm going to show you the structure. Maybe I'll show it actually. So this is the structure of a protein called chymotrypsin. And chymotrypsin starts out as a single polypeptide that's cut into 3 pieces and put back together. And I've colored the 3 separate pieces in different colors here. And the active site I've labeled in purple and red, but we don't need to worry about that. But what we can see is, like, there's quite a lot going on there. It's a little bit hard to see, like, can you see any secondary structure in there? It's very hard to see what's going on there. If we draw out all of the atoms, which is what this is basically doing at each of these vertexes and the ends, there's an atom. So, maybe an oxygen or a carbon or whatever it might be, or a hydrogen. So what a Richardson diagram does, which on this they call a ribbon, no, cartoon, is it overlays and it shows us where those are. And what we can do is we can hide those side chains, and it allows us to quickly see where the secondary structure is. And now we're like, oh, this has a single alpha helix and a couple of different clusters of beta sheets. So the power of the Richardson diagram was it gave a way to go from that quite complex structure to showing this, which is very easy to follow with the eye. What it doesn't give you a good concept of is the space, okay? And if we want to see so if we show the sticks, that gives us a better idea of what space this actually takes up, and it shows us the orientation of the side chains. But if we want to see what it really looks like, we would need to do a surface diagram. And so this shows us what the molecule actually looks like, okay, in 3-dimensional space, okay? Any questions about that? Okay, so that's where the exam is going to go up to. Are there any questions? No, so far? Okay, we'll introduce the idea of domains. And again, it's probably something I'll return to in the next lecture. Domains historically trip students up. That's not why I introduced them. They're complicated, but it turns out very important. For third and fourth year classes, if you go into cancer biology or cell signaling, you really need to understand domains. And we've learned through trial and error that if I introduce it to people in second year, it actually, and then we reintroduce it in third year, and then you get it again in fourth year, people start to understand it. So the idea with a domain is that a single protein, a single polypeptide, might actually be folded up into multiple functional units. Okay? And so in this example, we have a single protein that has what we would call two domains. And the way to think about this, now it's key to understand this is all in a single peptide. There's secondary and tertiary structure, but when we look at the tertiary structure, in this example, this tertiary structure actually looks like it's made up of 3 different proteins, if you know what you're looking for. Okay? And so it kind of looks like this, what's called an SH3 domain. And this is a domain that binds to prolines. It has this SH2 domain in blue, which binds to what we call phosphorylated tyrosine residues. And then in orange and yellow, it has this functional catalytic activity, which looks like a protein kinase. It's all on a single polypeptide. The idea here is that we are really complicated, and signaling pathways especially become very complicated. And for these pathways to evolve would be very, very complex, and it would take a very long time for these pathways to evolve. If we imagine that proteins can click together different units, okay? We imagine they can click together different units. That's how this domain theory works. So I'm part of a protein, I'm a domain, and what I do is I write my name on everything. Doug, 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 everywhere I go, okay? And I swap in this domain, and what this domain does is this domain grabs onto this box specifically. So what do? I grab this box, and I write Doug all over it. Okay, people following that so far? Here I'm the Doug domain, but instead of a domain that grabs this box, I have a different domain, and that one grabs umbrellas. So what's going to happen to this poor umbrella? If this is your umbrella, come up to the front and claim it at the end of class. What's going to happen? I'm going to write on this, right? Doug, Doug, Doug. So instead of 2 really complex proteins evolving, one that evolves to write all over this alcohol swab box, and another that separately evolves to write all over umbrellas, what if genetically I could just move bits around? And so we're not a genetics class, and I'm not asking you to understand the genetics at this point, but it turns out that actually that can occur. So parts of genes can be copied and moved around. And it's actually fairly straightforward, and this happens genetically reasonably commonly. And so what this is allowed for, if you're staying with me, is really complex proteins and really complex pathways have evolved in a relatively short time frame. Okay? Because this unit that grabs onto alcohol wipes, if it's attached to a domain that writes on them, it's going to grab them and write. But that same domain that grabs alcohol wipes might be bound to another domain that crushes the boxes. you see? And so by moving them around, we can change the functionality. You might want to think of it as like my laptop here. So my laptop does the computer part, but I can, through this cord I've attached to the camera at the back of the room, I could disconnect that. I have a mouse, I can connect that or disconnect it. I could connect in another camera. We're plugging in different parts in rather than building, okay, I'm going to build a computer that has a mouse and can project. So I design and build a computer to do that. Now I want one that has a drawing board, but I don't need a camera, so why don't I make a brand new computer for that? Okay? So by just swapping in parts genetically, we can build these complicated proteins. The key, though, remember, is that it ends up being a single gene that makes a single polypeptide that when it folds up, actually maintains those domains. And you can predict where those domains are using software. And so the reason that we love this, if we're researching a complicated pathway, is that if we know the linear sequence of a protein, so we can sequence the DNA of a gene, I've identified this gene that's mutated in cancer. I have the linear sequence of that, so I know the sequence of the amino acids. If I know the sequence of amino acids, I can run it through programs to predict what domains there are. And so by looking at this protein, if I didn't know anything else about it, I'd say, oh yeah, it binds tryptophan, phosphorylated tyrosines, and it's a kinase, so it adds a phosphoryl group to the substrate it binds to. So you can make predictions about the function, which is really important for researchers. Evolutionarily, that's why this has happened, but it's a great tool for researchers. Yes? You can do this synthetically, yeah. You can do anything synthetically now, basically, but yeah, when you can easily do this. And I don't know if I think I have an example of doing that at the very end of the course. And the back?
So each separate section like a functional protein on its own, or do they only one particular one?
Yeah, if you took scissors and you cut between those domains, they would actually stay folded and functional. Yeah. Theoretically, yes. This picture has three. The green one, the blue one, and then the orange and yellow one is actually a single domain. Oh, they just colored it in so you can see the boundaries of the domains. If you stretched that out, you'd see it be a single line, and parts of it would be light blue, parts of it would be green, parts orange and yellow. That's just used for identification. The dark green is just showing linker regions. Regions that link the domains together. Okay, so I think we'll end there, and we'll come back to this in the next class.